Everyone, today I'm going to be comparing S. aureus and MRSA, and I'm going to be highlighting some key characteristics about each bacterium. So we're going to be going over the structure of both bacterium, their mechanisms of action, and the mechanisms of resistance for the respective bacterium. So let's start by going over the characteristics of S. aureus and MRSA. We're going to start with S. aureus. So S. aureus, or Staphylococcus aureus, is a bacterium that lives in the nasal passages and on the skin of about 30% of people, and it normally does not cause infection. S. aureus, however, can be pathogenic under certain conditions. Commonly, if there is a weakening of the immune system, S. aureus can become pathogenic and cause an infection for someone. This attribute makes S. aureus an opportunistic infection. Moving on to MRSA, or methicillin-resistant Staphylococcus aureus. This is a strain of S. aureus that has acquired resistance to methicillin, a beta-lactam antibiotic. This resistance makes MRSA more difficult to treat than S. aureus because we can't use the same antibiotics that we would use for S. aureus for MRSA. Notably, MRSA infections commonly occur in people who were recently in the hospital or another healthcare setting. When this occurs, we refer to this as healthcare-associated MRSA. And then for the similarities for S. aureus and MRSA, so as we can assume, because MRSA is a strain of S. aureus, they are pretty similar in structure. S. aureus and MRSA are both gram-positive bacteria, meaning that they have thick peptidoglycan cell walls. This will become really important when we talk about antibiotics that are used to treat them and how those antibiotics work to treat the infection. Also, both bacteria have a grape-like structure. This is implied by the Greek suffix coccus, which means grape. So in the picture there's a sort of visual of what S. aureus or MRSA would look like under a microscope, a bunch of clusters of circular bacteria. So let's move on to the genomes of S. aureus and MRSA and the mechanisms associated with each bacterium. For S. aureus, the genome of S. aureus possesses several virulence factors. This includes genes for exotoxins and enzymes such as coagulase. So these virulence factors are going to help the bacteria evade the immune system and cause damage to the host tissue. So for S. aureus infections, penicillin is commonly prescribed to combat this infection. So penicillin is a beta-lactam antibiotic, and its mechanism of action involves the inhibition of bacterial cell wall synthesis. Penicillin specifically targets PPBs, or penicillin binding proteins, that play a role in peptidoglycan synthesis. So when this happens, the cell wall becomes weakened and it lyses. So this goes back to what we discussed on the last slide, talking about how S. aureus and MRSA both have thick peptidoglycan walls. So penicillin is going to inhibit that cell wall synthesis and weaken the cell wall to stop the infection from spreading. Moving over to MRSA, the most prominent feature of MRSA is its MECA gene. So this gene encodes for PB2A. That might sound familiar from the PBP we talked about with S. aureus. But in MRSA, this penicillin binding protein is actually what causes the methicillin resistance in MRSA. So MRSA's PPB2A gene has low affinity for those beta-lactam antibiotics such as penicillin that we talked about. So this results in the continuation of MRSA's cell wall synthesis because the beta-lactam antibiotics are not effectively able to bind. And so the bacteria cannot stop the infection from spreading. So if we look at the visual down there, for S. aureus, we see that the penicillin is easily able to bind onto the PPB within the cell wall, but in MRSA, we see the PBP2A and its binding site is a different shape, so penicillin can't effectively bind on. So this is how penicillin is able to resist beta-lactam antibiotics. And this is why MRSA then requires a different family of antibiotics to be effectively treated. So to conclude, S. aureus is a bacterium that is non-pathogenic unless there's a compromise in the body's defenses. MRSA is a strain of S. aureus that is resistant to beta-lactam antibiotics, such as penicillin. MRSA's resistance to beta-lactam comes from its MRSA gene, which codes for a penicillin binding protein that has low affinity for beta-lactam antibiotics. So this inhibits the antibiotic's mechanism of action and allows the MRSA infection to continue.